Hello, everybody. Welcome to Thursday, January 7th, 2021. It's a great day to be a junior Potter. Mr. Parrott here with your science lesson for Thursday. First off, answer up that bell ringer for today. I'm going to keep on reminding you of that so we get in the habit of doing those again. We're going to look at what DNA does to make more DNA. We talked about this happening in mitosis, that replication has to happen, that DNA has to make a copy of itself. Well, how does it actually do that? We're going to take a look at how DNA actually does that for real today in our notes, and there's a couple of steps that it has to go through. So we're going to take a look at that, and then there's a little activity at the end that I'm going to have you create your own DNA model in a slideshow, something that I'm kind of proud of that we put together. It took me a while to get it to function correctly, so hopefully everything works out for us here with it. So let's look at what we've got going on here. We went Thursday, DNA replication notes. So this is just all the assignments. Your notes, go back to Wednesday and you'll be able to pull up the notes for today. I posted them on there. We're on number 2 now. So for where you'll find the notes are in the Wednesday section. I'll probably bring them over into the Thursday stuff as well. So they'll be in the Thursday folder by the time you get this. I'll probably put it right there, put just what the notes are there that we're doing number 2 there. I said there a couple of times. I need to stop doing that. Number 2, looking at replication. So first off, why do we need more DNA? Why can we not be satisfied with the DNA that we have already? First off, we need DNA to replicate itself because we need 2 copies for mitosis, because when you take one cell and boop, make it into 2 cells like we talked about earlier in the year, each one of those new cells needs to have a copy of DNA to do mitosis with. Without that information there, one of these cells, if not both of these cells, would be mutated cells. And mutated cells are not necessarily good for your body. They're not going to give you superpowers. You're not going to shoot laser eyes out of your eye sockets with it. You're not going to have adamantium claws coming out of your knuckles. You're not going to be Wolverine or Cyclops or any of those things. Usually what it leads to is some kind of genetic disease or a genetic disorder is what we call them, which are not things that we particularly want to have. They're not going to be good for you. So usually we do not want mutations to occur. Sometimes they're good, but a lot more times they're probably going to be bad for us because it's not our way of our body functioning in and of itself. So that's why we need to replicate it, to make sure those new growing cells has a copy for it. And DNA is just important in general because it is going to make everything for our body. It's going to be the instructions of how to make stuff, which we'll get into that more over the next couple of weeks of how it's going to actually be making those things. The steps to replication, I got it boiled down to 4 steps. The first thing with DNA is that big old double helix, the twisted ladder. So it's doing this thing. First thing it needs to do, here I'll stop doing it, it's all twisted up like this. Think of it like my arms. First thing it needs to do is it needs to unwind itself, so it's just 2 straight lines, and then it needs to unzip like a zipper and split itself in half. You can see that in that first picture up there, right underneath of me. Where's my camera? Right underneath of me there. It needs to split itself up, and it unwinds and splits kind of at the same time as it's going through there. So it's going to unwind, unzip. That exposes the bases. You can see the little teeth coming off of it there. Those are the bases. What happens is that a new DNA base, a new nucleotide, comes in to pair up with that free end. So you can see that happening in the middle. I got the arrow pointing to it. Our little GIF picture here. We've got these free ends coming here. It takes a nucleotide and just attaches it to it. So it uses one side as like the blueprint, and it just comes in and matches up with what it needs to pair up with. Now I know what you're thinking. How does it know that it needs to do that? I talked about it in yesterday's notes where there's like a lock and key system where one connection needs to have 2 keys, whereas one needs to have 3 keys. If they don't have the appropriate amount of keys, it triggers something in the DNA to know that it's wrong, so it goes back and fixes and corrects their mistake there. What then happens is that once it recognizes that that is bonded together, that is connected together in the way that it needs to, it recognizes it's the correct pair, it bonds together. Not only do the bases bond, but the backbone will bond as well. The sugars and the phosphates will connect up. So there's two different bonds happening. When everything is cemented up, it begins to curl itself back up and rewind it, making one out of one end and one out of the other. So you can kind of see that as well in this picture where one new part goes to the left, one new part kind of goes off to the right, so it kind of will split, and a new one will get made, and a second new one will get made. Each of it having half old DNA, half new DNA, which is kind of a cool little thing too, because it uses one side as the blueprint, so it's always going to have one of the previous strands there with a brand new strand going on with it. And they're just molecules, so it's not like they're going to age out. That's why we're able to have that here. You've got a video that kind of will go through these stages of replication for us. It's about an 8 or so minute video. It's a real good video that walks us all the way through. It's the Amoeba sisters going through there. They'll talk about a little bit about DNA, so it's good for DNA just to get updated, and then it goes through those steps of replication and really breaks it down for us. Because that's all of our notes today. Very quick notes on what's happening here. I know it might not seem like very quick after you get through my explanation and you watch the video, but it is pretty quick depending on other things that we do. Your assignment then is back on in the Thursday folder for today. We got this replication assignment, and you're going to open that up. It is a slides kind of thing here. So here's our replication activity. You can put your name where it says name here. We got our directions. It says at your desk there will be a DNA sequence with a number on it. For you at home, I have that in the Thursday folder. You have this document that says replication, transcription, edible DNA activity, DNA sequences document. You need to open this up. And all you need to do, there's going to be a bunch of these sequences, as shown here. There's 12 different ones. You just have to pick one of them to do. So if you want to do number 1, all you got to do right here where there's this green part, write down that you chose number 1. If you want to go with 10 for whatever reason, you can go with 10. All I did was create 12 random codes for you so that not everybody's doing the exact same one, so everybody's doing a little bit something different. On the next page, you're going to have a key to what everything represents, because it might not be as you might not be able to see all of the wording on here, so just to make sure that you know what things are what, I got a little color key here. We got oranges for the sugar, purples for the phosphates, and then our different colors for our different nitrogen bases. What you're going to do is you're going to assemble a model of DNA. And how you're going to do that, you're going to go back to the sequence. Let's say we had sequence number 1 here. It's AGCTA. What you're going to do is going left to right. You're going to make a model of DNA. So the first thing that we're going to do, we're going to start off with a sugar. You're then going to attach a phosphate to it because it's always a sugar and a phosphate. And you can see that these puzzle pieces kind of just fit right over the top of each other. So you can click and drag these on your iPad to have it fit right over the top of it. Boom. There's 2 pieces right there. Actually, this would be up top. So you first start with the top strand is first. And then what you're going to do is, let's see, what was our first thing? An A. Well, here's our A right here. Here's our nitrogen base. That would be the first thing that we have going there for us. We then need to create the next pair. So what connects to a phosphate? Well, that would be another sugar. Fits right in the little hole there with our puzzle piece. We get another little phosphate on top, a little connector piece. Second piece, we have an A and a G. Well, here's our G. Notice that on some of them, you might have to flip it around. How to flip it around, you'll see this little thing, the little circle on it. That's how you can rotate it. So you might have to rotate it to get it to fit in that nice little square peg. All of them are like puzzle pieces, so if something doesn't seem quite right, just flip it around, because you're going to have to do that when you make the second strand, because we're going to do replication. Let's just stop right there for right now. We're going to look at the second half of it. We'll start off with some DNA. We're going to have to rotate it around, so there's our rotation. We'll have to rotate the phosphate around. Boop. We'll connect it up there. Now, you're going to have to think to yourself and remember, what is our base pair that is going to pair up with an adenine? Well, if you remember what we talked about yesterday, A's go with T's. So you'll take your T, boom, connect it up, and then you can make it look actually nice and fit together. Hey, there we got a section of DNA. You'll continue on doing that with our next piece. Well, this one was a G. What partners up with a G? Well, that'd be our C here. And I have to rotate that one around too. Boom, there we go. A G and a C are connected up together again. Each one of these is only 5 bases long, so you're only going to have to do 5 of it. So you're not going to do like an incredibly long tail. But essentially it is going to demonstrate not only us making the process of replication, because you're going through the process of replication because you're starting out with one strand and finding what it pairs up to make the other side. That's basically what replication does. It also works on knowing our base pairs. What thing goes with what in there. So once you get your model assembled, there should be enough pieces. I made sure that there's enough pieces on there. You got a couple of questions to answer up at the end. You got 8 questions, 4 on one slide, 4 on the other side. As it says at the top, answer all the questions based off of our activity and our notes. You don't need to copy and paste from the web. You shouldn't have to. You should be able to answer all of these. Remember, if you are copying and pasting, it's wrong. It's going to be marked wrong. We're going to have some talks about a thing called plagiarism. So do not cheat. Do not just copy and paste those things there. So that's an example. If you want to choose one, because I already did a couple of it for you there, you have a good example to go off of. If you need any assistance whatsoever, let me know. Once again, this is the assignment for Thursday, January 7th, 2021 there. Do it tomorrow, so if you need any help, let me know. They're going to have some time in class there to work on it after we're done going through the notes and watching our video. And I show them kind of how the assignment works with it, so they're going to be piecing things together themselves in here. But that's all for today. I hope you're doing well, and I will see you back here real soon. Always remember, it's a great day to be a junior potter. See ya.